extreme weather can have is threatening our food supplies. Today we're here with Emily Wengler, and she's a crop scientist from University of California, Davis. She's going to explain to us how we can potentially fight this food crisis by learning about the plant's defense mechanisms against the insect herbivores. Thank you for being with us, Emily.
Thank you for having me.
So can you explain how we can combat this food crisis by learning about the plant's defense mechanisms?
Yeah, so we are working towards solutions to feed a growing population in the face of a warming climate. We will need to produce about 70% more food by 2050 to be sustainable, and when the average temperature increases by 1 degree Celsius, we lose about 10% of crop yields. By studying how plants defend themselves, we have found that we can mimic the molecules that stimulate plant defense, and this way we can produce more resilient and effective crop yields.
So you're saying that we can induce the plant's defense mechanisms against the insect herbivores in order to make them more resistant?
Yeah, so we have found that elicitor molecules released from insect saliva are actually recognized by plants to initiate a defense response against specific insects.
Emily, do the plants have different defense mechanisms?
Yeah, so they have the first line of defense, which is deterring characteristics like spines, thorns, and trichomes. And then there are secondary metabolites, which are used to deter the insects. There are constitutive secondary metabolites, which are always present in the plant. And then there are induced secondary metabolites, which is my specialty, which are present after a specific compound triggers a response.
You mentioned the induced secondary metabolites that help with the plant defense. If they help with defense, why doesn't the plant just produce them all the time?
It is important for plants to only use these molecules when necessary because it takes energy away from growth and reproduction, which could affect their survival as well as crop yields.
So let's get a little into the cellular and molecular level of plant defense. With animals, when an animal is injured, the cells of the immune system become activated to regenerate that tissue and also to fight off the pathogens. Is that the case with the plants?
So plants are a little different because the cells are encapsulated in a cell wall, which has a rigid structure, so they're not able to mobilize to the site of infection. So they all have to be competent to initiate a response.
Thank you so much, Emily. We have here with us Skylar Russon, a professor of plant physiology at University of California, Davis. Skylar is going to explain to us about the genes and pathways that are involved in plant defense against insect herbivores. Thank you for being with us, Skylar.
Yeah, thank you.
So can you start by explaining how the plants even recognize that they're being wounded by the insects, and how do they respond to that?
Yeah, absolutely. So plants have evolved to recognize specific elicitor molecules that are present when they've been wounded by an insect. And this allows them to differentiate between mechanical damage versus when they're under attack by another organism. And studies have shown that with Arabidopsis, grasshopper oil secretions that are applied to the leaves of the plant initiate unique changes in signaling as opposed to mechanical damage to the leaves alone. OPDA is believed to be the key elicitor molecule that signals to the plant that it's under attack by grasshoppers, and it's actually a membrane lipid that's released by the grasshopper lipase. So after the plant recognizes it's under attack by an herbivore, calcium is released into the cytosol of plant cells. And calcium is released from organelles that store it, such as the ER, mitochondria, the vacuole in the cell wall. And after the concentrations of calcium in the cytosol go up, regulatory proteins are activated, including calcium-dependent protein kinases and an important protein called calmodulin. Once calmodulin becomes active, it's able to bind and activate an important transcription factor in Arabidopsis called IQD1. And the IQD1 transcription factor is then able to activate glycosinolate synthesis. And experiments with Arabidopsis has shown overexpression of this transcription factor actually yields plants that are more resistant to insect herbivores. Glycosinolates are small organic molecules that are secondary metabolites, and they have a range of effects on insects that can be repellent to toxic.
So, Skylar, do you know of any specific examples of glycosinolates in plant cells?
Yeah, sure. One example would be the cabbage family of plants, including mustard plants. And mustard plants produce these glycosinolate molecules that are always present in the leaves. And then when the leaves are damaged, such as by an herbivore, they are mixed with an enzyme and they produce these really reactive isothiocyanate molecules that are toxic to insects. And isothiocyanates are also responsible for flavors of plants we enjoy, such as mustard, radish, and wasabi.
So I'm seeing here that you've also mentioned MAP kinases. Is that also involved in plant defense?
Yeah, so the MAP kinase pathway has been shown to be very important to plant defense, and in Arabidopsis, application of the grasshopper oral secretions elicits unique changes in this pathway. MAP kinase initiates the chain of phosphorylation that leads to the activation of MAP kinase protein, which can then in some plants activate specific defense genes, and it also activates jasmonic acid biosynthesis. Jasmonic acid is an important plant hormone that regulates many aspects of plant defense. And the hormone is synthesized in the chloroplast, where linoleic acid is enzymatically converted to OPDA, which is then transported from the chloroplast to the peroxisome, where OPDA is finally converted to jasmonic acid, which is released into the cytosol.
So you're saying that at the end, jasmonic acid is going to be accumulated in the cytosol? How's that going to affect anything?
First, it stops plant growth altogether, which is important because the plant needs to spend its available energy in producing secondary metabolites for defense rather than primary metabolites. The other thing jasmonic acid does is it activates defense genes. And for a lot of these defense genes to be activated, jasmonate needs to be conjugated to an amino acid to be in the bioactive form. When jasmonate is conjugated to isoleucine, it's able to form an active E3 ubiquitin ligase with other proteins that can then bind to the Jaz protein repressor and target it for ubiquitination of the proteasome. This allows the MYC2 transcription factor to initiate transcription of defense genes.
Okay, so the defense genes, do they help with the protection of the plant?
Yeah, they do. So there's two main categories of defense genes. They can activate toxic or repulsive secondary metabolites like we talked about before, but they can also encode defense proteins that inhibit herbivore digestion. A few examples of those are amylase and proteinase, which block starch and protein digestion, as well as lectins, which block nutrient absorption by the organism. I think this is one of the most interesting topics in plant defense. And what happens is the plant recognizes the elicitor molecule that we talked about, and it actually releases a cocktail of secondary metabolites as gases. And these are referred to as herbivore-induced plant volatiles. They can do a number of things, including attracting natural predators of the insects that are harming them. They can repel other herbivores, and they can even warn nearby plants of the threat. And so in experiments done with corn plants, the elicitor molecule of elicitin from armyworm saliva has been shown to stimulate the synthesis of herbivore-induced plant volatiles. And quartz seedlings treated with elicitin release volatiles that attract parasitoid wasps that then harm those predators.
Thank you again, Skylar. It's always nice to have you on the show.